7.0 Molecular genetics, from DNA to proteins. The spiral structure in the picture is a large organic molecule. Can you guess what it is? Here's a hint: molecules like this one determine who you are. They contain genetic information that controls your characteristics. They determine your eye color, facial features, and other physical attributes. What molecule is it? You probably answered DNA. Today, it is commonly known that DNA is the genetic material. For a long time, scientists knew such molecules existed. They were aware that genetic information was contained within organic molecules. However, they didn't know which type of molecules played this role. In fact, for many decades, scientists thought that proteins were the molecules that carry genetic information. In this chapter, you will learn how scientists discovered that DNA carries the code of life. 7.1 DNA and RNA. Lesson objectives. State the central dogma of molecular biology. Outline discoveries that led to knowledge of DNA's structure and function. Describe the structure of RNA, and identify the three main types of RNA. Vocabulary. Central dogma of molecular biology. Chargaff's rules. Messenger RNA, mRNA. Ribosomal RNA, rRNA. Transfer RNA, tRNA. Introduction. Your DNA, or deoxyribonucleic acid, contains the genes that determine who you are. How can this organic molecule control your characteristics? DNA contains instructions for all the proteins your body makes. Proteins, in turn, determine the structure and function of all your cells. What determines a protein's structure? It begins with the sequence of amino acids that make up the protein. Instructions for making proteins with the correct sequence of amino acids are encoded in DNA. The vocabulary of DNA, chromosomes, chromatids, chromatin, transcription, translation, and replication is discussed at this link. 1823. Central dogma of molecular biology. DNA is found in chromosomes. In eukaryotic cells, chromosomes always remain in the nucleus, but proteins are made at ribosomes in the cytoplasm. How do the instructions in DNA get to the site of protein synthesis outside the nucleus? Another type of nucleic acid is responsible. This nucleic acid is RNA, or ribonucleic acid. RNA is a small molecule that can squeeze through pores in the nuclear membrane. It carries the information from DNA in the nucleus to a ribosome in the cytoplasm and then helps assemble the protein. In short, DNA-RNA protein. Discovering this sequence of events was a major milestone in molecular biology. It is called the central dogma of molecular biology. You can watch a video about the central dogma and other concepts in this lesson at this link, 807. An overview of protein synthesis can be viewed at this link, 1046. DNA. DNA is the genetic material in your cells. It was passed on to you from your parents and determines your characteristics. The discovery that DNA is the genetic material was another important milestone in molecular biology. Griffith searches for the genetic material. Many scientists contributed to the identification of DNA as the genetic material. In the 1920s, Frederick Griffith made an important discovery. He was studying two different strains of a bacterium, called R. rough strain and S. smooth strain. He injected the two strains into mice. The S strain killed, virulent, the mice, but the R strain did not, non-virulent. Griffith also injected mice with S strain bacteria that had been killed by heat. As expected, the killed bacteria did not harm the mice. However, when the dead S strain bacteria were mixed with live R strain bacteria and injected, the mice died. Griffith's experimental results. Griffith showed that a substance could be transferred to harmless bacteria and make them deadly. Based on his observations, Griffith deduced that something in the killed S strain was transferred to the previously harmless R strain, making the R strain deadly. What was that something? What type of substance could change the characteristics of the organism that received it? Avery's team makes a major contribution. In the early 1940s, a team of scientists led by Oswald Avery tried to answer the question raised by Griffith's results. They inactivated various substances in the S-strain bacteria. They then killed the S-strain bacteria and mixed the remains with live R-strain bacteria. When they inactivated proteins, the R-strain was deadly to the injected mice. This ruled out proteins as the genetic material. Why? Even without the S-strain proteins, the R-strain was changed, or transformed, into the deadly strain. However, when the researchers inactivated DNA in the S-strain, the R-strain remained harmless. This led to the conclusion that DNA is the substance that controls the characteristics of organisms. In other words, DNA is the genetic material. You can watch an animation about the research of both Griffith and Avery at this link. Hershey and Chase seal the deal. The conclusion that DNA is the genetic material was not widely accepted at first. It had to be confirmed by other research. In the 1950s, Alfred Hershey and Martha Chase did experiments with viruses and bacteria. Viruses are not cells. They are basically DNA inside a protein coat. To reproduce, a virus must insert its own genetic material into a cell, such as a bacterium. Then it uses the cell's machinery to make more viruses. The researchers used different radioactive elements to label the DNA and proteins in viruses. This allowed them to identify which molecule the viruses inserted into bacteria. DNA was the molecule they identified. This confirmed that DNA is the genetic material. Chargaff writes the rules. Other important discoveries about DNA were made in the mid-1900s by Erwin Chargaff. He studied DNA from many different species. He was especially interested in the 4 different nitrogen bases of DNA: adenine, A, guanine, G, cytosine, C, and thymine, T. Chargaff found that concentrations of the 4 bases differed from one species to another. However, within each species, the concentration of adenine was always about the same as the concentration of thymine. The same was true of the concentrations of guanine and cytosine. These observations came to be known as Chargaff's rules. The significance of the rules would not be revealed until the structure of DNA was discovered. The double helix. After DNA was found to be the genetic material, scientists wanted to learn more about it. James Watson and Francis Crick are usually given credit for discovering that DNA has a double helix shape, like a spiral staircase. The discovery was based on the prior work of Rosalind Franklin and other scientists, who had used X-rays to learn more about DNA's structure. Franklin and these other scientists have not always been given credit for their contributions. You can learn more about Franklin's work by watching the video at this link, 747. The double helix shape of DNA, together with Chargaff's rules, led to a better understanding of DNA. DNA, as a nucleic acid, is made from nucleotide monomers, and the DNA double helix consists of 2 polynucleotide chains. Each nucleotide consists of a sugar, deoxyribose, a phosphate group, and a nitrogen-containing base, A, C, G, or T. The sugar-phosphate backbone of the double helix was discussed in the Chemistry of Life chapter. Scientists concluded that bonds, hydrogen bonds, between complementary bases hold together the 2 polynucleotide chains of DNA. Adenine always bonds with its complementary base, thymine. Cytosine always bonds with its complementary base, guanine. DNA replication. Knowledge of DNA's structure helped scientists understand how DNA replicates. DNA replication is the process in which DNA is copied. It occurs during the synthesis, S, phase of the eukaryotic cell cycle. DNA replication begins when an enzyme breaks the bonds between complementary bases in DNA. This exposes the bases inside the molecules so they can be read by another enzyme and used to build 2 new DNA strands with complementary bases. The 2 daughter molecules that result each contain 1 strand from the parent molecule and 1 new strand that is complementary to it. As a result, the 2 daughter molecules are both identical to the parent molecule. The process of DNA replication is actually much more complex than this simple summary. You can see a detailed animation of the process at this link, 205. RNA. DNA alone cannot tell your cells how to make proteins. It needs the help of RNA, the other main player in the central dogma of molecular biology. Remember, DNA lives in the nucleus, but proteins are made on the ribosomes in the cytoplasm. How does the genetic information get from the nucleus to the cytoplasm? RNA is the answer. RNA versus DNA. RNA, like DNA, is a nucleic acid. However, RNA differs from DNA in several ways. In addition to being smaller than DNA, RNA also consists of one nucleotide chain instead of two. Contains the nitrogen base uracil, U, instead of thymine. Contains the sugar ribose instead of deoxyribose. Types of RNA. There are three main types of RNA, all of which are involved in making proteins. Messenger RNA, mRNA, copies the genetic instructions from DNA in the nucleus and carries them to the cytoplasm. Ribosomal RNA, rRNA, helps form ribosomes, where proteins are assembled. Transfer RNA, tRNA, brings amino acids to ribosomes, where they are joined together to form proteins. In the next lesson, you can read in detail how these three types of RNA help cells make proteins. Lesson summary. The central dogma of molecular biology states that DNA contains instructions for making a protein, which are copied by RNA. RNA then uses the instructions to make a protein. In short, DNA-RNA protein. The work of several researchers led to the discovery that DNA is the genetic material. Other researchers discovered that DNA has a double helix shape, consisting of two polynucleotide chains held together by bonds between complementary bases. RNA differs from DNA in several ways. There are three main types of RNA: messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA. Each type plays a different role in making proteins. Lesson review questions. Recall. State the central dogma of molecular biology. Outline research that determined that DNA is the genetic material. What are Chargaff's rules? Identify the structure of the DNA molecule. Why is DNA replication said to be semi-conservative? Apply concepts. Create a diagram that shows how DNA replication occurs. Think critically. Explain why complementary base pairing is necessary to maintain the double helix shape of the DNA molecule. Compare and contrast DNA and RNA.